All right, I apologize to the audience. There's a lot of me in a row, but all right, so welcome to Designing the Future, Safer and Effective Psychoactive Compounds for Mental Health. So classic compounds like psilocybin in its natural form will always have a home in the psychedelic medicine space. But the future of the industry will be shared with next generation drugs, new discoveries and variations that could improve experiences and outcomes for patients. So we have some experts in that subject here. We'll go down the line. Let's start at the end with some introduction a little bit.
Hi everyone, my name is Cher. I'm the co-founder and CEO of CB Therapeutics. We're a biotech company that has engineered strains of yeast to make different molecules such as cannabinoids, psychedelics, and now looking into longevity molecules as well.
Good afternoon. I'm Dr. Amy Reichelt. I'm the Chief Innovation Officer at PurMinds Neuropharma, and we are developing psychedelic and non-psychedelic molecules for the treatment of neurological conditions.
And I'm Sam. I'm the founder and CEO of Terran Biosciences. As my background, I did my undergrad degree in neuroscience at MIT and my MD and PhD at Columbia. I founded Terran in 2017 to help develop new therapies for patients with mental and neuropsychiatric disorders. And now Terran is one of the largest manufacturers of GMP psychedelics that we provide at no cost to researchers and clinics worldwide, as well as a number of other new chemical entities we've designed to improve upon both classic and novel psychedelics.
Let's stay on that, Sam. I don't think we touched much on that in the previous panel about your production and your development. Maybe a little bit more on that. Yeah.
Sure. So Terran is a GMP manufacturer of psychedelics for clinical use, as well as non-GMP for preclinical use. And we manufacture at large scale psilocybin, LSD, MDMA. And we provide compounds like this to researchers and clinicians for free, at no cost to them, worldwide. We have manufacturing plants in Europe and are mass producing these compounds, and also to support our future 505(b)(2) applications to help bring generic drugs to the market as well. And then, separate from that, we have a number of labs in the UK that we've worked with to design new psychedelics. And we've actually designed over 10,000 new psychedelics and then produced a number of them. And I'll give you an example. We've now produced new forms of both classic, like psilocybin, LSD, DMT, the world's first orally active DMT, the world's first long-acting MDMA. But we've also improved some of the rarer psychedelics. We have new versions of a compound called 2CB. We've improved across the empathogens with new versions of MDAI, MBDB, MDE, and methylone.
And can I add to that as well? So PurMinds Neuropharma, we're based in Toronto. We have a production facility as well that is focusing on our botanical psilocybin. We have been growing our psychedelic mushrooms in-house and are in the process of developing a new way to be able to extract the psilocybin using our proprietary technology at in-house to be able to have a quantifiable botanical psilocybin that will be able to be supplied to clinical trials or therapeutic uses in the future. So that's something that has been emerging in the last year within PurMinds.
And I think it's a great panel that you've organized because we're all working on novel psychedelics in some way, but we all have a very different approach. So it's very cool to have and meet everybody and discuss this. So our approach is a little different. We're primarily focused on the synthetic biology side of things, more on the sustainability. So if you think about any psychedelic that's being made, one of the things you have to ask yourself is, once there is FDA approval, when these things are in the market at a larger scale, how sustainable and cost effective would something like that be? So those are the sorts of questions that we're sort of tackling with the yeast engineering and other platforms that we're working on.
Maybe a little bit more specifics on that, on your angle of going about that?
Yeah, and so I guess the angle that we go for is we identify specific genes in different sorts of plants or fungi. We put those genes through our own proprietary platform to enhance the production, enhance the stability. For a lot of these genes, they may not be as functional or as stable when you remove them from the original plant or the fungus. So part of the challenge is to be able to make those genes robust. The other challenge is if you look at a particular molecule that may be toxic to a living organism, the question is how do you engineer enough of that molecule within that organism before it dies? So there's different things that strategies that we've taken, sort of like transporting the molecules outside of the cell before, as we're continuing fermentation to allow for higher throughputs as well.
And hopefully discovering something interesting and doing something with it. I mean,
Yeah,
maybe a bit about your development pipeline now, or some more.
So we have a few clients in the psychedelic space, and we are developing not only the original psychedelics for them. We've actually developed over 60 some molecules for them, but now we're looking into analog libraries. So for example, one of our customers wanted a library of 200 of a particular type of psychedelic. So part of the original challenge is the yeast may be engineered only to make that natural molecule. So how do you make an efficient way where now you can integrate some sort of technology into the yeast itself, where now it can go out there and give you different analogs at will, right?
Maybe a little bit more context on what you're working on, Amy, at PurMinds. A little bit more.
Yeah. So we have multiple pathways at the moment that we're pursuing in terms of our pipeline. So as I was saying, that we have both our Pure 001, which is our proprietary botanical formulation, which will be maintaining the tryptamine profile with the entourage effect, which has been shown to be important in terms of the therapeutic potential of psilocybin, and knowing as well that the move to legalize or decriminalize aspects of psilocybin for therapy within clinical spaces has currently focused around the botanical aspect. We feel that it's really important for us to be able to have a standardized way of extracting the molecules from the psychedelic psilocybin-containing mushrooms. We also have our own clinical pipelines that we're progressing. So we have both psychedelic pipelines, where we're using existing psychedelics, but in combination with other modalities or chemicals that are able to augment neuroplasticity, which is a fundamental aspect of the therapeutic mechanism of psychedelics. And also NCEs, where we're focusing on a non-hallucinogenic molecule at the moment. Again, focused around neurological conditions, which are very much underserved, particularly in the psychedelic space at the moment, where the majority of clinical trials are focusing on major depressive disorder, PTSD, other psychiatric conditions. I think that there is, an important niche and an important need for drugs that can help therapeutically with neurodegenerative conditions and neurological conditions, which go hand in hand with people's quality of life and well-being, but really harnessing the potential of these compounds to regenerate neurons through augmenting neurogenesis or boosting brain-derived neurotrophic factor that can then enhance synaptic plasticity and formation of new connections in the brain. And I think that this rewiring is an aspect of the therapeutic potential of both these molecules for psychiatric conditions, where people are looking at changes to their ways of thinking, but then also in terms of therapies for neurodegenerative diseases, where the brain's inherent ability to rewire is helping to overcome changes that are happening in terms of neuronal degeneration.
No, definitely. On that, Sam, I think you had mentioned earlier one of your developments was around an LSD blockers, a different kind of category of development. Maybe you want to, I don't think we spoke about that much last panel. Maybe talk about that.
Sure. So that gets into the idea that you can take a psychedelic, a classic psychedelic that's been shown to be safe and effective, and combine it with a selective serotonin 2A blocker to remove the trip. Now, this comes out of some inventions from a number of academics in Europe and the United States. And they asked the question, they said, we look at psychedelics and they hit a lot of receptors in the brain. But we also know that only one receptor is responsible for the trip, the serotonin 2A receptor. What would happen if we take a psychedelic which is hitting all those receptors, combine it with a serotonin 2A receptor blocker, remove the trip, would it maintain the therapeutic efficacy? And they did that in a number of different preclinical studies, and it did. It was just as effective without the serotonin 2A effect. But at Terran, we took it a step further, because as we know, scientists have already cured all diseases known to mouse kind. Doesn't always translate to humans. And so we did this data analysis study with our collaborators at Yale, from a study in Europe, where they had done this MRI imaging study, where patients got a baseline MRI scan. Then they took LSD, 100 micrograms. They got another MRI scan a week later. And that showed all the functional rewiring and functional connectivity of the brain. And those maps you see with all the connectivity changing. But then, we waited another week. They got a serotonin 2A blocker and LSD. No trip. No change on the scales of functional perception overall. They went in the scanner, and amazingly, LSD was doing the same thing, rewiring the brain in the background. And that, for us, was the aha moment. That without the serotonin 2A receptor, you can remove the trip by combining a psychedelic with a 2A blocker, but maintain the psychedelic's effect on neuronal rewiring. And that's one of the things we're developing. We partnered the serotonin 2A blockers, exclusively licensed from Sanofi, who transferred to us 15,000 patients' data, 100 clinical trials, and 5 INDs. And now we're developing that forward as a potential take-home medication to maintain the therapeutic benefit of the psychedelic and potential benefit on neuroregeneration of the brain, so a direct application and longevity without the trip.
And to add to that as well, what I think is really important is that we know that these compounds, molecules, are able to exert this sudden surge in neuroplasticity, but it's what you do with that neuroplasticity that's the most important thing. If you have a dosing of your psychedelic molecule and put your brain in this plastic state, but then remain in your habits that are detrimental to your well-being, then you're just reinforcing those habits in an even more plastic state. So things are just going to become more ingrained. That's why the integration is so important in following a psychedelic-assisted psychotherapy. But then also, when it comes to neurological conditions, this post-treatment where you want to use physiologically relevant stimuli to entrain these brain pathways of relevance to be able to overcome potentially neurodegenerative conditions or encourage the rewiring in the right way. It's like when you dismantle the radio, you've got to put it all back together in the right way.
Definitely, yeah. You don't want to go on Twitter right after a trip. You want some healthier integration. On the removing the trip aspect, so there's been studies on that, but there is some sort of argument in the space that the trip, there's some benefit to it, perhaps therapeutically. I guess you're speaking neurologically, that the effects are the same in terms of potential benefit at the neurological level?
Well, and that's a great question. So to answer this question, we need to look at how we treat these mental health conditions right now. And I think that's where we're going to find the answer, and I'll give you an example. Post-traumatic stress disorder. The patient is going along. They're happy, their life is good, and then a traumatic event happens. And after that, they go downhill. We already know giving drugs alone doesn't work as well as giving the actual psychotherapy because you're dealing with that traumatic life event. So you need that psychotherapy. And psychedelic-assisted psychotherapy is incredibly effective. And that's one example of where the trip might be one of the most important aspects. However, we also know for just normal major depression. Most of the time, the patient doesn't report any specific life event. They're just going along, and over periods of time, they become more depressed, and they report anhedonia, and they can't point to a specific event. And we already know that we're already able to treat that with SSRIs, with MAO inhibitors, with SNRIs, without therapy. Depression already responds to drugs alone without therapy. The problem is the drugs take months to work. They have severe potential. Sometimes they have severe side effects. Sometimes they can cause sexual dysfunction and weight gain and other issues. But they do ultimately work in many cases. And what we are trying to do for patients is to harness the power of psychedelics for those patients so they could have a take-home psychedelic that'll act rapidly, hopefully without the same side effects as the SSRIs and the other ones on the market. So a new first-line therapy, which we think would have greater access for patients who can't afford to take time for a full trip or don't want a full trip, or maybe they can't handle a full trip, or they have a background of a psychotic illness. So in that sense, for us, it's all about offering patients more options. So you can meet the patient where they are. For patients that want the trip, you can offer that. For patients that can't take the trip, you give the tripless psychedelic.
And importantly as well, by eliminating aspects of the trip, you're making the drug become more accessible, but also diminishing the stigma associated with a psychedelic. For many people, I mean, this also applies to potentially military veterans who've been told never to take drugs, or individuals of generations that feel that they have been told these will fry your brains, and you'd be turning back that stigma clock by saying, look, we're using these as scaffolds. They're the blueprint to then be able to make these drugs that are better, safer, can be taken in your own home. They have less chance of divergence and abuse potential because there's no risk involved in having a trip, or then having to drive somewhere, or you'd feel that you're able to work that day. You won't have to take the afternoon off after your psychedelic treatment. So I think that the importance is opening the accessibility to more and more people, and then the acceptance across the board.
Yeah, definitely.
Yeah, I think it's fascinating. Just 5 years ago, this sort of talk would be almost impossible if you think about it from a perspective of legality and where the medicine and how doctors and people perceived it. So I think it's a great time to be in this industry, especially at the forefront where you're able to make novel molecules that may not have psychoactivity. And again, there may be a lot of different types of people that may not want that effect. So I think it's the golden era. And I think the one thing that does concern me a little bit is there may be too many, molecules out there, and how do you, parse through all of these, let's say, millions of potential analogs to come down with the right one that may have the right effect. So I think that is probably something that's going to take a little bit longer with the clinical trials and whatnot. But having said that, it's a great thing to have more molecules than less, right? So.
Yeah, I mean, you're all working on your own libraries of development. Maybe a little bit staying on that, the comparison of how traditional SSRIs work in the brain to these potential blocked non-hallucinogenic. What would be the difference in mechanism of action? One of the experts to say, because SSRIs have some efficacy, it's not the highest percentage. So how would they differ?
Oh.
I could comment on that first.
Yeah.
So the big issue is SSRI stands for selective serotonin reuptake transporter. That's the transporter that's controlling the reuptake of serotonin. So by blocking it, you're getting kind of a global flood of serotonin at all the spots, and it's generally activating a lot of different serotonin receptors. At the same time, we know psychedelics not only act on serotonin receptors, they act on dopamine receptors with LSD, norepinephrine receptors, and that recent paper that just came out directly binding the TrkB receptor for BDNF. And so there are a lot of other highly targeted targets, and it may be either the direct target or the symphony of targets it's acting on. Whichever it is, we're seeing that psychedelics seem to have a much more rapid and lasting effect than SSRIs.
And importantly as well, that I think that the 2A receptor is highly expressed in the cortex. And that is the key area of the brain, particularly the prefrontal cortex, which is involved in executive function, decision making, and being able to control your behavior. And being able to exert plasticity in this area of the brain when plasticity becomes diminished through synaptic loss, as is seen in individuals with, for instance, major depression, that replenishment of plasticity in these key areas for cognition are what's going to help people so rapidly see the effects of the therapy and the efficacy of these psychedelic and psychedelic-inspired drugs. I think that it's like a laser-focused target, and also the rapidness of these drugs and how people are reporting changes and shifts in mood really at the cessation of their trip. They're coming out and they may have had a challenging experience, but yet the 24 hours later they feel better. Whereas with an individual who you've just put onto an SSRI, 72 hours later, they're probably complaining that their gut hurts and suffering from the peripheral side effects. And then, if it is seen to be successful, may also be experiencing a plethora of side effects, such as loss of libido and emotional numbing. So it's so important that these drugs are so rapidly acting, but also showing this durability. And what's been really exciting in terms of the clinical trials is seeing efficacy in the measures 3 months, 6 months afterwards.
Definitely. So it seems what we're learning here, it's not either or. It's not remove the trip or you absolutely need the trip. And it seems from what I'm hearing about Taryn is offering patients a variety of options. It's not one size fits all. Someone may want to go deep and have a difficult experience and come out of that with a difficult trip. Somebody who's maybe a bit more hesitant or can't fit, and maybe that doesn't scale. So you remove all that. So it seems as if we want to get patient access. We want to increase options, which is what all this development is about, to provide a bunch of options for the different patient populations.
And another thing as well is that with the design of the novel molecules, we can start to eradicate off-target effects such as 2B receptor agonism, which has implications particularly around cardiovascular and heart valve thickening. And you don't want to get cardiac valvopathy. And by removing that aspect of the drug, you're reducing the risk and therefore opening up the access of that drug to a broader population.
And one of the items I want to get into then to answer that question is what we did with empathogens. Empathogens are like MDMA, for example. But MDMA is very powerful and might be too powerful for some people in certain settings. And so we were developing another compound and we chose a variety, MDAI, MBDB, MDE, and we also were developing one called methylone. And here's where we came across a really exciting discovery, which was that, interestingly, when it dosed at a full effective dose, it produced the same sort of empathy as MDMA, but it's reported to be more mild. But the exciting thing is that when dosed at a lower dose, it can potentially produce anxiety. And we wanted to understand this and say, why does half the dose produce anxiety? You would think half a dose would just be a more mild experience, but it's actually producing the opposite. So we studied the molecule and we said, look, the molecule exists as a mixture of two forms, the left-handed side and the right-handed side. It's a mirror image of each other in equal amounts. It's called enantiomers. So we split the molecule and purified left-handed and right-handed forms. And we found in preclinical models that all the anxiety at the low doses was produced by the left-handed form. And by removing that completely and only having an enantiomerically pure right-handed form of the molecule, at any dose, it would just produce different dose-dependent effects without the paradoxical anxiety in the preclinical models. So we're very excited to be making those breakthroughs in empathogens, making the molecules safer.
Yeah, that is, a low dose does tend to give some anxiety. Removing that would be very interesting, yeah. And I believe the root word for left-handed was sinister, so you're removing some of that. Yeah, all right. Sherry, you had something?
No, this sounds very exciting, what you're doing, and it's very interesting to see that there are different sorts of outcomes based on just the enantiomers. There's definitely precedents in cannabinoids with the same thing and other molecules, so it's good to see that there's being work done in this side as well.
Yeah, while you have the mic, we're wrapping up. I mean, things that maybe you're excited about your research right now, looking forward?
So I think there's a few different things, but just to keep it short, we are at a point where we are going to introduce some new families of psychedelics, and we're gonna, we just filed a patent, so that information will be out soon. We are working on a range of other new molecules and now expanding into longevity, things like resveratrol, curcumin, etc., developing that through the bioinformatics platform, NAD+. So we're really excited about introducing new molecules, finding new molecules that have a therapeutic or nutraceutical value, and being able to recreate the supply chain from the back end.
Very interesting. Amy, closing thoughts?
I think that it's an exciting field and an exciting time to be able to address a number of conditions that have previously been seen as being too complex to treat because of mixed etiologies that are involved in the pathology. So one of the beauties of the psychedelics is that they have these multi-targeted approaches and that you're hitting numerous targets that can then all work together to be able to help attenuate the neurodegenerative process or neurological conditions or or depression. So I think that this is a really exciting place to be in.
Sam, any...
We're really excited to be at the intersection of psychedelics and longevity. And we think that for the first time in history, the combination of a psychedelic with a two-way blocker to remove the trip enables the psychedelic, which has been shown to have longevity producing neuroregeneration, to now have applications in the longevity space as opposed to only the antidepressant mental health space. And so we're really excited to bring that forward, to really push forward a neuroregenerative in the longevity space to address what we see as studies have shown after age 40, 5% per decade brain shrinking. And we think with a psychedelic with no trip, that could address that and bring more treatments forward for longevity.
Great, that's a great ending. I appreciate it, Sam, Amy, Cher. That was a great panel. Thank you very much. All right, thank you, audience.
Bigger applause for these panelists.